Hello, welcome to QAxon. In this video, we will be seeing about thyroid gland, the hormones secreted by it. We are going to see in detail how the hormones are formed and their actions. Thyroid gland is an endocrine gland which is situated at the root of the neck on either side of the trachea. It has two lobes which are connected in the middle by the isthmus. Histologically, it contains two types of cells called follicular cells and parafollicular cells. We have thyroxine, triiodothyronine, and reverse T3. These hormones are secreted by the follicular cells. And we have got one more hormone called calcitonin. It is secreted by the parafollicular cells. These parafollicular cells are also called as C cells. Now let's see the different steps involved in the synthesis of thyroid hormone. First step is the formation of thyroglobulin. It is synthesized from tyrosine in the thyroid follicular cells. Next step is the iodide pump, which is going to pump in the iodide into the cell. Then the iodide is oxidized to iodine. The next step is the organification of iodine, which is followed by coupling reaction. This coupling reaction results in the formation of monoiodotyrosine and diiodotyrosine. Remember that each step in the synthesis is stimulated by thyroid stimulating hormone, which is secreted from the anterior pituitary gland. Now let me explain the steps involved in synthesis of thyroid hormone. We have the thyroid follicular cell, and the thyroid follicular cell has a basolateral side and an apical side. Inside the thyroid follicular cell, we have the thyroglobulin, which is formed from tyrosine in the rough endoplasmic reticulum. Along the basolateral side, we have the sodium iodide symporter. It is otherwise called sodium iodide cotransporter. This helps in iodide trapping. Iodide trapping is the process by which iodide is pumped into the thyroid follicular cells. The iodide has to be converted to iodine. This takes place in the presence of hydrogen peroxide and the enzyme peroxidase. This iodine that is formed is pumped into the apical side through the chloride iodine pump. It is called pendrin. You can see here the pendrin is located on the apical side. The iodine now, which is on the apical side, combines with the thyroglobulin. So this process is called organification. Now we have MIT, that is monoiodotyrosine, DIT, diiodotyrosine, which are formed as a result of organification. One molecule of DIT and MIT combine together to form T3. Two molecules of DIT combine to form T4. This thyroglobulin is still attached to all the molecules that have been formed. Now this molecule, that is thyroglobulin, which is still attached to all the molecules that have been formed, they enter into the thyroid follicular cell through a process of pinocytosis. Now this molecule is then digested by proteases, and T3 and T4 are released along with MIT and DIT. These hormones T3 and T4, by the process of exocytosis, are secreted to the basolateral side. Now this MIT and DIT undergo deiodination in the presence of the enzyme deiodinase, and tyrosine is released, which is again used by the endoplasmic reticulum for the formation of thyroglobulin, and this process repeats again, and this results in the formation of thyroid hormones. Now the formed thyroid hormone has to be transported. It is transported being bound to thyroid binding globulin, thyroxine binding prealbumin, and albumin. Moving on to regulation of thyroid hormone secretion. Hypothalamus secretes thyrotropin releasing hormone which acts on anterior pituitary and results in the secretion of thyroid stimulating hormone. This thyroid stimulating hormone acts on the thyroid gland and this results in the formation of T3 and T4. Please note that T3 and T4 they have a negative feedback mechanism on the anterior pituitary gland. Now let's see the mode of action of thyroid hormone. In the target cells, T4 is deiodinized to T3. So the T3 that has been formed combines with the thyroid hormone receptor which is placed in the nucleus. So it is either attached to the DNA material or in very close proximity to it. So apart from this receptor, we have another receptor called retinoid X receptor. Now T3 when combined with this receptor forms what is known as hormone receptor complex. This complex initiates the transcription process by activating the enzymes such as RNA polymerase and phosphoprotein kinases. There is a large number of mRNA that is formed and these mRNA activate the ribosomes to synthesize new proteins. These proteins are involved in many activities like in the cardiovascular system, the hormone can cause increase in cardiac output, increase in heart rate and increase in respiratory rate. In metabolism, there is increase in number of mitochondria and mitochondrial activity. There is increase in sodium potassium ATPase activity, increase in oxygen consumption, glucose absorption, lipolysis, gluconeogenesis, and there is increase in BMR. Thyroid hormone is very essential for CNS development. It also plays a big role along with growth hormone in growth of the individual. These are some actions of thyroid hormone.